So good morning. It gives me great pleasure to introduce David McConkey to you this morning. David was one of my mentors in the lab when I was a fellow at MD Anderson. So I got an opportunity to learn a lot from David. He's primarily a cancer biologist who has been commandeered into the Department of Urology to contribute mostly to bladder cancer research and has done a lot of high-impact stuff, particularly recently. He did his PhD at the Karolinska Institute in Stockholm, and then moved on to Dana-Farber for his postdoc, and then has been at MD Anderson for a long time, and despite his young years has been full professor for an awfully long time. But anyway, I'd like to invite David up to tell us something brand new that has everybody talking in the bladder field, to talk about intrinsic subtypes of bladder cancer.
Thanks, Peter. And as a hockey player, it's good to be back in hockey country again. And actually, you're very lucky, You're very lucky to have Peter, because we loved him and we tried to keep him, and he escaped nevertheless. So he's a really special guy. So you probably all know this, but we are pretty pathetic in terms of our management of bladder cancer. We really haven't made much progress in a very, very long time. The frontline therapies for this disease have remained the same for decades, and we don't have good biomarker tests to help us determine prognosis at this point. Bladder cancer is really considered two diseases, non-muscle invasive and muscle invasive, and there's plenty of preclinical work to suggest that they progress along two different tracks. The papillary or non-muscle invasive pathway tends to be associated with activating mutations in FGF receptor 3 and maybe RAS pathway activation, whereas the non-papillary or muscle invasive pathway tends to be associated with loss of tumor suppressors, P53, P10, and RB. P10 usually isn't genetically deleted, but it's often epigenetically silenced in bladder cancer. The good news is that many of us were involved in a project called the Cancer Genome Atlas Project, where we obtained very, very deep molecular information about 128 muscle-invasive bladder cancers. And this paper was just published a couple of months ago in Nature. I'm not going to go through the details of that work, except to say that this is really tour de force, in-depth characterization. It's not just about bladder cancer, but it's actually being done on 25 different forms of cancer. And everything we can think of to do to these tumors is being done, from the protein all the way down to the DNA level. So what this means is the future for bladder cancer, and for that matter, any of the other cancers that is being studied by the TCG and other programs, looks bright. But now's the time, I think, when we're going to start to see substantial rapid impact being made in bladder cancer and other solid tumors because of this genomics work. So our near-term priorities are ambitious, and I'm not going to talk about our programmatic moonshot plans, which is something we were charged to do by our new president at MD Anderson. But I will kind of outline some of the high points here for what we're trying to do in our program. So, number one problem is to determine how many cancers we're actually dealing with here. We want to understand the biological heterogeneity of bladder cancer as deeply and as thoroughly as possible, because we know these cancers have highly variable clinical courses, and we believe that these variable clinical courses are caused by differences in biology. And then in the very near future, we want to be able to distinguish good cancers from bad cancers, and which cancers are going to respond to our therapies, our current therapies, which actually work pretty well in half the patients with respect to chemotherapy, and most of the patients with respect to BCG. But we can't predict in whom they're not going to work. And it would be very helpful for us clinically to know that, because chemotherapy is still underutilized in muscle-invasive bladder cancer. And finally, a longer-term goal is to use this deep biological information to develop targeted agents in bladder cancer. We don't have any FDA-approved drugs that hit biological targets in bladder cancer as of right now. So we need to fix that problem quickly. I'm going to give you a brief introduction about breast cancer, because one of my take-home messages today is that bladder cancers look a lot like breast cancers. And an investigator named Chuck Peru, who's at the University of North Carolina, used whole genome mRNA expression profiling about 15 years ago, and unsupervised analyses, meaning ignorant of the clinical outcomes in this cohort, to identify what are now household name subtypes of breast cancer. People call them intrinsic subtypes. And you may all be familiar with these, but in case you're not, they're called luminal A and luminal B, which are the two subtypes that contain estrogen receptor and are treated with tamoxifen. Basal-like, which you'll hear more about from me today. HER2, many of these tumors contain amplified ERB2. And then finally normal-like, which some people now group together with the basal-like tumors, and others believe may be sometimes contaminated from normal breast tissue. And what's interesting about these subtypes is that they express biomarkers that are shared with cells at different stages of differentiation within the differentiation hierarchy of the normal breast. So the normal-like, or we sometimes call them Claudin low tumors, as well as the basal tumors, in my opinion, I've drawn an X through the arrow that was in the original review, are both very similar in appearance at the biomarker level to the normal breast epithelial stem cell. Whereas luminal A, luminal B, and HER2 tumors are more similar to the more differentiated cells within the normal breast, ductal epithelial cells. So aside from all this biology, what has been the clinical impact of knowing this? It turns out that these different subtypes really behave as separate disease entities, and clinically they're managed very differently. So, as I mentioned earlier, if you have a luminal A or a luminal B breast cancer, you're managed with endocrine therapy. If you have a HER2 or triple-negative breast cancer, triple-negative cancers tend to be basal, these agents don't work. There's no activity. On the other hand, if you have a HER2 or triple-negative cancer, you will obtain benefit from cytotoxic chemotherapy, particularly in the neoadjuvant setting. However, cytotoxic chemotherapy does not work well in luminal A breast cancers. Why is that? Well, as it turns out, these intrinsic subtypes respond differently to neoadjuvant chemotherapy. So this is another study by Chuck Peru where he showed, using our data from MD Anderson, that the so-called normal-like Claudin low, the basal-like tumors, and the HER2 tumors all have fairly high rates of pathologic complete response in response to pre-surgical chemotherapy. But if you look at the luminal A tumors, they really don't respond well at all. There was a 0% path CR rate in those tumors. Luminal B is mixed. About 20% of those tumors responded to chemotherapy. And why is this? Well, if you look at the right side, this is a gene expression signature associated with proliferation. So as it turns out, the basal-like cancers have very high proliferation rates. Claudin low and HER2 are more intermediate. The luminal A's are very low. So there's a correlation, very strong correlation, between proliferation and subtype in breast cancer. And it's prognostic. So if you have high Ki67, you're more likely to have a basal cancer or a HER2 cancer, and you're also more likely to die of your disease in the absence of chemotherapy. So that's this blue curve right here. However, given neoadjuvant chemotherapy, your survival curve is up here, along with the luminal A and luminal B cancers. So most of the benefit from neoadjuvant chemotherapy in breast cancer is obtained from the triple-negative basal breast cancer subtype. And this is associated with high proliferation rates. Proliferation, as you know, tends to be associated with chemotherapy sensitivity. This has created something that's called the triple-negative paradox. So on the one hand, having a triple-negative or a basal cancer is associated with poor clinical outcomes in patients who are not treated, managed with neoadjuvant chemotherapy. But exactly the same biology is associated with good response to chemotherapy. So that's the paradox. Bad intrinsic biology, but chemosensitive. So as Peter mentioned, we just used the same kind of approach. Oop. we just used a very similar approach to the one that Chuck Peru used in breast cancer to characterize the heterogeneity in muscle-invasive bladder cancers. So what we did was we pulled 73 TURs, fresh frozen tumors, ignored any clinical outcomes data, performed whole genome expression profiling, and used unsupervised hierarchical clustering to look at the tumor's heterogeneity. What we decided was that we could see 3 separate disease subtypes. And you can see them here in the red, the green, and the blue. And the red subtype was associated with worse outcomes in this cohort. Being cancer biologists, we were actually looking at the biomarkers, the top biomarkers that were associated with the membership of these tumors within each of these 3 subtypes. And we recognized immediately that the red subtype was full of biomarkers that were shared with basal breast cancers. Mostly high molecular weight cytokeratins, but also CD44, peak adherin, c-MYC, just canonical breast cancer biomarkers. In fact, if we take a breast cancer basal gene expression signature and apply it to these tumors, we see strong correlations in gene expression between these tumors and basal breast cancers. On the other hand, the blue subtype was loaded with luminal breast cancer biomarkers, including CD24, cytokeratin 20, GATA3 or B2 or B3. All of the really canonical breast cancer luminal biomarkers were enriched in these tumors. And again, taking a luminal breast cancer gene expression signature, we could see this was very similar to luminal breast cancers. And then we confirmed this at the protein level by immunohistochemistry. These markers are mutually exclusively expressed. So if you have a basal cancer, you're full of basal markers, but no luminal markers, and vice versa. So we also used some tools to look at what was responsible for the gene expression signatures in these subtypes. And satisfyingly, we rediscovered something we'd already been working on in the basal subtype, namely a protein called P63, which is a marker of the basal layer of the epithelium in essentially all epithelial tissues, prostate, skin. And so P63 is known to be also a stem cell marker, and it's also known to control the biology of basal breast cancers. So this was very satisfying and confirmed for us that the basal phenotype in these tumors was caused by P63. On the other hand, when we looked at the luminal subtype, what we discovered was that it was enriched for peroxisome proliferator activator receptor gamma, or PPAR gamma activation. Now, PPAR gamma is not associated with luminal breast cancers, but it is thought to be implicated in risk of developing bladder cancer. There are studies suggesting that use of anti-diabetic drugs that activate PPAR gamma increase the risk of getting bladder cancer. So again, this was consistent with what we already knew about the biology of bladder cancer. And finally, this green cluster was enriched for an active P53 gene expression signature, which I'll tell you about more in a minute. So we used some bioinformatics tools to confirm that these gene expression signatures were actually caused by these transcription factors. I won't go into the details, but we generated so-called gene signatures in human bladder cancer cell lines by modulating these transcription factors, and we showed that these gene signatures were enriched in the basal or luminal cancers respectively. So we know that the gene expression in the basal cancers in patients is caused by P63, and we know that the luminal gene expression signature in luminal bladder cancers is caused by PPAR gamma. That was important to us to demonstrate this cause-effect relationship. Now, in parallel, a colleague of mine and a close friend, Billy Kim, at the University of North Carolina, discovered more or less the same thing. So in a paper that came out very close to ours, when ours came out, that he published in PNAS, demonstrated that bladder cancers could be divided into basal and luminal subtypes that look just like breast cancers. He decided that he could see 2 subtypes, and we decided that we could see 3, and I'll tell you why in a moment. Finally, as part of our Cancer Genome Atlas project, we discovered the same thing in the TCGA data. So using RNA sequencing, which is a more accurate tool to measure RNA expression than we use by genomic expression profiling, and unsupervised cluster analysis, we decided that there were 4 subtypes present in the TCGA dataset. and that cluster 3 was enriched with basal biomarkers and clusters 1 and 2 with luminal biomarkers. And importantly, as I'll tell you about in a minute, cluster 3 was enriched with squamous histopathologic features. So we took the TCGA data and we compared our subtypes to the ones that were identified by Billy Kim's group and by the TCGA, and they overlap very, very closely. So if you look here at the top, Billy's tumors here are indicated basal in red and luminal in blue, and essentially all of our basal cancers are contained in his basal subtype, and almost all of our P53-like and luminal subcancers are included in his luminal subtype. Similarly, the TCGA tumors are arranged here. You can see here's the cluster 4, cluster 3, cluster 2, and cluster 1 from the TCGA. And again, you can see by eye there's remarkable overlap between the TCGA calls and ours. And again, looking at the basal and luminal markers here, you can see that we're all enriched with basal markers on this side and luminal markers on that side. And again, this is using the TCGA data set and our different classifiers to assign the tumors to subtypes, and then comparing the subtype assignments at the end. So why do we think there are basal and luminal cancers? The simplest explanation, just as I showed you for that hierarchy of differentiation in the breast, is that basal cancers derive from the basal layer of the urothelium, whereas the luminal cancers derive from either the umbrella cell layer or some other more differentiated layer of the urothelium. So they retain biomarkers from the state of differentiation at which they were transformed. We actually really are attracted to this idea, and it suggests that looking for one bladder cancer stem cell or one mouse model of bladder cancer is a mistake. At the very least, we have to consider the possibility that there are two different origins of bladder cancer here, basal and luminal. So because we noticed the similarity to breast cancer, we performed some other studies. The take-home messages here are that the P53-like tumors, these green tumors in our cluster diagram, look a lot like luminal A breast cancers. And I'll remind you, luminal A breast cancers are the ones that tend to be very resistant to neoadjuvant chemotherapy. Similarly, the basal cancers that we'd identified in bladder were very similar to the basal breast cancers. Parenthetically, the same kind of analysis has been done in other tumors. And prostate cancers, actually, the majority of them look like luminal A breast cancers as well. This isn't terribly surprising if you consider the fact that P63 loss is often used diagnostically to identify prostate cancer. So prostate cancers, unlike bladder cancers, appear to be almost entirely luminal in origin. Now, whether neuroendocrine tumors are basal or not is something I think could be explored in the future. But this is an important distinction. This is probably why androgen receptor targeting works so uniformly well, even in men with very advanced disease. These cancers are still addicted to androgen receptor because they're luminal. So what is it about these P53-like tumors that makes them distinct from the other luminal cancers? They all have luminal biomarkers in them. Biologically, what's different? So I said they have an active P53 gene expression signature. What that means is that they tend to have lower proliferation rates. So like luminal breast cancers, these cancers tend to be more quiescent. And this is shown here using the gene expression data that I mentioned earlier. You can see the green here corresponds to the low levels of cell cycle and proliferation genes that are present in these tumors. They have another feature that we think is really important, and that is that they're enriched with myofibroblasts, cancer-associated fibroblasts. So this is showing you some of the top genes that identify this subtype. Essentially, all of the top 10 genes associated with this molecular subtype are myofibroblast genes. So the cause-effect relationship here is interesting to consider. Are the fibroblasts in there causing this P53-ness, or is it just that the P53-ness is attracting them? We don't know that at this point, but right now we're isolating fibroblasts, cancer-associated fibroblasts, from primary cancers to try to understand what they look like and what they do. Mattias Höglund is a pathologist working at the University of Lund in Sweden, and he had identified his own classifiers looking at intrinsic subtypes. And we compared our subtypes to his subtypes, and confirmed that one of the subtypes that he identified that he called infiltrated, that he showed were infiltrated with fibroblasts, corresponded well to this p53 subtype, consistent with this idea that one of the important features of their biology is that they're infiltrated with stromal cells. And then finally, looking at pathways, you can see that extracellular matrix genes, which are commonly associated with infiltrating fibroblasts, were also enriched in this p53-like subtype. So overall, we think low proliferation and stromal infiltration are what distinguish the p53-like tumors from the other luminal tumors. Now, what about TCGA clusters 3 and 4? Turns out we were working on another process that was relevant to this distinction. TCGA clusters 3 and 4 are distinguished by epithelial to mesenchymal transition. Now, EMT is a developmental process that gets reactivated during wound healing, where epithelial cells lose their normal cell-cell contact with each other and become more like fibroblasts, more migratory and invasive. And many investigators have demonstrated that this process gets activated in cancers to promote metastasis. In particular, there's an investigator at MIT named Bob Weinberg who's made a lot of really important contributions recently demonstrating the importance of EMT in metastasis. And again, this is from one of his review articles. Typically, tumor cells have areas in them that are exposed perhaps to hypoxia or maybe cytokines like TGF-beta or maybe inflammatory signals coming from the immune system or other sources. And these signals cause reversible changes in phenotype associated with EMT. The cells become less attached to each other, more invasive and migratory. They are capable then of entering the vasculature. And now if we isolate these CTCs, we can show that they express markers of EMT. They tend to associate with platelets, and the platelets reinforce this EMT in them. And then once they land at the distant site, the whole thing reverses itself. They undergo a process called mesenchymal to epithelial transition, where they lose the mesenchymal markers and invasiveness and acquire more of an epithelial phenotype. Now, why do they do this? Again, it seems to be about proliferation. So proliferation and invasion seem not to go on at the same time. So in order to become invasive, tumor cells turn on EMT, but turn down proliferation. When they hit the distant organ site as metastases, if they turn down EMT, they can turn up proliferation again. So we did an analysis of clusters 3 and 4 because we were suspicious that this might be going on and confirmed that cluster 3 is enriched with P63. We've done gene signature analyses to confirm that. And cluster 4 is enriched with a protein called ZEB1, which drives EMT. And so as a consequence of that, cluster 4 is enriched with mesenchymal markers and cluster 3 with epithelial markers. Again, Billy Kim's group at UNC recognized this as well. They recognized that one of the subsets of basal cancers looks a lot like Claudin-low tumors, Claudin-low breast cancers, which others had shown were characterized by EMT. So you can see over here are the Claudin-low tumors. They tend not to express high levels of epithelial cytokeratins, but they do express high levels of EMT markers. And so as a result of all of this, and this is where I'm going to finish the descriptive part of this talk, this is what we think the subtypes actually look like. So Billy Kim was right in identifying 2 subtypes of bladder cancers, basal and luminal. But we were able to subtype the luminal cancers further by virtue of this expression of low proliferation and stromal infiltration. So this is our P53-like subtype, which is a luminal subtype that's contained within his luminal subtype. Then finally, the TCGA designations were also correct, because the TCGA discovered this EMT that distinguishes 2 subtypes of basal cancers. So overall, there are 2 luminal subtypes and 2 basal subtypes of bladder cancer. And down here at the bottom, you can see that they correspond very well to the subtypes of breast cancer. So what does this mean clinically? We know something about what's going on with these cancers, but we still have a lot to learn. So basal cancers tend to be more prevalent in women. They're enriched with squamous and sarcomatoid features. I'll show you the data for squamous in a moment. So in other words, these are not separate disease entities, as maybe we thought previously, but squamous and sarcomatoid features are associated with basal cancers. If you see them, it's almost diagnostic that this is a basal cancer. Basal cancers, as I mentioned, are associated with shorter disease-specific and overall survival in the absence of chemo. But as I told you about in breast cancer, in the presence of chemo, things are changed. So first, the squamous features. Our fellows noticed immediately that the basal cancers were enriched with squamous features from our discovery cohort. And so in subsequent cohorts, we looked for them again, and we found them consistently associated with basal cancers in multiple independent cohorts. The TCGA discovered the same thing in their data set, and Billy Kim discovered the same thing in his data set. Bogdan Cherniak, our pathologist, did IHC on a big TMA that was created by an international bladder cancer consortium. And what we found again was that expression of cytokeratins 5 and 6, which are basal biomarkers, and cytokeratin 20, which is a luminal biomarker, is mutually exclusive at the protein level. Tumors tend to have one or the other, but not be double positive. And looking at cytokeratin 5-6, tumors with cytokeratin 5-6 were enriched with squamous differentiation, whereas tumors without cytokeratin 5-6 but with cytokeratin 20 were not. Multiple independent data sets, including all of ours, this is one of them, Billy Kim's, and finally Matthias Hergland's data set, indicate that in the absence of effective chemotherapy, these tumors are lethal. And this isn't surprising, given what we know about basal breast cancers, plus the fact that they tend to be enriched with EMT. These tumors are intrinsically nasty. They have high proliferation rates, and they're highly invasive and metastatic if given the opportunity. And again, just to emphasize the proliferation rates here, you can see the red here are the same cell cycle genes I was pointing out earlier. They're highly enriched in these basal tumors. They're also characterized by high c-MYC levels, just as basal breast cancers are. Now, Yair Lotan's group, Yair Lotan is a urologist at UT Southwestern, has already done a lot of work on the relationship between cell cycle and proliferation and prognosis. And we knew in bladder cancer that high Ki-67 levels are associated with poor outcomes in the absence of chemotherapy. This is one of the papers he published in Clinical Cancer Research in 2006. And this is a follow-up study that he published with colleagues in JNCI just a few years ago in 2009. So again, using Ki-67, you can actually get pretty good prognostic information about this lethal phenotype. So like breast cancer, neoadjuvant chemotherapy is active in bladder cancer. There's level 1 evidence to support its use. Unfortunately, utilization rates are still below 20%, unless people are paying attention. So in our SWOG 1011 trial, we've been doing a surgical trial to look at the difference between extended and standard lymph node dissection and outcomes. And because everybody's paying attention, utilization rates are over 50%. So when people are really worried about it, we're using chemotherapy, but in general practice, it's being used at a rate of under 20%. And that needs to change. The reason for that is that because of another SWOG study that was led by Bart Grossman's group, we know that the benefit from neoadjuvant chemotherapy tends to be modest at 5 to 10% impact on overall survival. So there's concern that you're delivering a pretty toxic regimen and deriving little benefit from it. Randy Milliken, one of my colleagues at MD Anderson, working with Chris Logothetis, performed a study demonstrating that it's not bad to delay surgery because of neoadjuvant chemotherapy. And it's also not bad to delay chemotherapy to give surgery first. You get identical outcomes either way, whether you operate first or whether you give chemotherapy first. So pre versus post, the outcomes are identical. So that gives us an opportunity to use neoadjuvant chemotherapy as a platform to try to understand response rates better. So Dan Willis, one of our fellows, again, looking through the data in our discovery cohort, noticed that all of the p53-like tumors were resistant to neoadjuvant chemo. So we expanded our neoadjuvant cohort and obtained an old one from Randy Milliken's work that I just alluded to, and looked at it in 2 more cohorts, and again, there was enrichment for resistance in this p53-like cohort. We took the p53-like gene signature out of these tumors, out of these primary tumors, applied it to our cell lines, and identified a subset of these cell lines that also were enriched with the same signature. And these p53-like cell lines were resistant to cisplatin-induced apoptosis. So in other words, this signature correlates with chemoresistance. It's not caused by wild-type p53 itself, we think. Looking at mutation rates in our cohorts, in the Swedish cohort, and in the TCGA cohort, these tumors are p53-like not because they have wild-type p53, but for other reasons that we're still trying to understand. So then we obtained another cohort from a collaborator of ours at Fox Chase, Betsy Plimak. This was a clinical trial of dose-dense MVAC where we had both pre and in many cases post-treatment tissues available. And again, we discovered the same relationship where we saw this chemoresistance in patients with these p53-like tumors. But given that we had the post-treatment tissue available, we were then able to compare the gene expression profiles pre and post. And what's worrisome is that a lot of the pre-treatment luminal cancers switched to become p53-like after being exposed to chemotherapy. And essentially, comparing the pre and post gene expression signatures in everybody and asking what's different after chemo, we discovered a new active p53-like signature that was active in all of them. So over here you can see pre and post, pre and post, pre and post. You can see that every time a tumor was exposed to chemotherapy, at cystectomy there was enrichment for this active wild-type p53 gene expression signature. So what are the implications? Well, the implications are that by delivering chemotherapy, if we're not successful at downstaging that tumor is resistant. It's p53-like after chemo. How long does that last? We don't know. But it certainly may explain why we're failing to have much impact with subsequent regimens after we failed with MVAC or GemCis. We may have to wait for this to dissipate or prevent it from happening in order for us to have effective therapy. So we attempted to identify biomarkers associated with chemosensitivity in each of these subtypes, given that the basal cancers and the luminal cancers, fractions of them were chemosensitive. We couldn't find a strong signature in the luminal cancers, but in the basal cancers, in 2 different cohorts, the chemosensitive tumors were enriched with lymphocytes. And this harkens back again to breast cancer, where it's been shown that breast cancers that are enriched with lymphocytes tend to be chemosensitive. Why? We don't know. Are they contributing to the chemo response? Possibly. Maybe it's partly immune-mediated that these tumors are responding. Alternatively, it may be just that the biology of these tumors attracts lymphocytes, and they're just correlated with chemosensitivity. The other thing, though, that's very important is that in this immune signature are things like CTLA-4 and PD-1 and PD-L1, which are markers of immune checkpoint activation. So one thing that we think is a very attractive hypothesis is that using immune checkpoint blockade, which is very commonly used now in prostate cancer and other diseases, we might be able to even further enhance chemo responses in these basal tumors. So I mentioned this triple negative or basal breast cancer paradox, that the same gene signatures that predict poor prognosis in the absence of chemotherapy tend to predict benefit from chemotherapy, and that this is associated with proliferation. We have preliminary evidence that the same thing is going on in bladder cancer. So here I'm sharing with you unpublished results from a neoadjuvant trial of dose-dense MVAC plus Avastin, which is a VEGF blockade. And what we found again was that the p53-like tumors were more resistant. But importantly, looking at survival outcomes, the basal cancers now are the best, and the p53-like cancers are the worst. So take-home message number one is that if somebody has a basal cancer, basal bladder cancer, that cancer definitely should be managed with neoadjuvant chemotherapy. No question about it. This is a potentially lethal and metastatic cancer that needs to be treated aggressively. And the benefit obtained is dramatic. Our results are actually anticipated, or were anticipated, by a reanalysis of the SWOG trial, where some colleagues of mine in the GU committee reanalyzed the data and correlated responses with presence or absence of squamous features. And again, most of the benefit from chemotherapy was in tumors with squamous features. The other take-home message is that if a patient has a p53-like tumor, it may be futile to use neoadjuvant chemotherapy on that patient. And this idea that quiescence is associated with chemoresistance is very, very common now throughout oncology literature. And in particular, if you go back to the breast cancer neoadjuvant clinical trials, you'll find plenty of evidence for an association between low proliferation rates and chemoresistance. And I'll just indicate one of these trials here. This is the I-SPY trial, which was a trial that we've modeled one of our own clinical trials after, where patients were treated with neoadjuvant chemotherapy, and the biology of those cancers was correlated with pathologic complete response rates. And what was discovered was that benefit from neoadjuvant chemotherapy was very modest in luminal A cancers and cancers that had wild-type p53 gene expression signatures, a lot like the ones that we've identified, and that most of the benefit was obtained in the triple-negative and highly proliferative tumors. And even in preclinical studies, this idea that wild-type p53 might not be a good thing for chemoresponsiveness is starting to emerge. So when I was a postdoc, I was trained to think that you needed wild-type p53 to undergo DNA damage-induced cell death. It's a part of the dogma these days. We're all taught this. But most of this work was done in lymphocytes and hematopoietic cancers that have trouble arresting in the cell cycle. They tend to have very rapid doubling times. They don't have as robust checkpoints in place. And so a colleague of mine at MD Anderson, Gigi Lozano, working again in a preclinical model of breast cancer, looked at this carefully and discovered that even the presence of one wild-type copy of p53 impairs chemoresponse because it allows the cells to become quiescent, to stop proliferating. And then they reemerge later to recur. So this idea that quiescence is really an important mechanism of chemoresistance is actually, there's plenty of evidence now emerging from the literature. So as I mentioned, we've designed a clinical trial to test a lot of this stuff prospectively. We had a workshop now, it seems like almost a decade ago, although it wasn't that long ago, where we discussed how we could exploit this opportunity in using neoadjuvant therapy to accelerate progress in bladder cancer clinical management. What we settled upon was this trial here, which is also known as SWOG 1314, where the primary goals here are really translational medicine goals. We're going to compare GemCis and MVAC head-to-head for the first time to see which one is better, if either. I think we're going to find that they're exactly equivalent and probably kill the same subsets of tumors. But then there is a huge discovery translational medicine effort going on. The primary goal is going to be to test a predictive algorithm that was developed by Dan Theodorescu, who's at the University of Colorado. But we're going to test our stuff. We've got BISQUIP funding to test our subtypes and the relationship between subtype and response to neoadjuvant chemo. And then Jonathan Rosenberg and others are going to be doing sequencing to see if there are mutations, DNA mutations that are associated with chemosensitivity or resistance. So this is going to be our opportunity to rapidly, because this trial should be opening within a couple of months, rapidly and prospectively test what I've just told you today. If it works, we're going to very aggressively push forward the idea that we need to subtype cancers and make sure that everybody with a basal cancer gets chemo, and perhaps that anybody with a p53-like tumor doesn't. We'll find something else for them. So what about the future? Well, in characterizing these subtypes, we noticed that they were enriched with biological features that might be therapeutic targets. So the basal tumors contain EGF receptors. They contain HIF-1 alpha, which might explain our Avastin data. They contain other things that can be targeted. The luminal tumors are enriched with activating FGF receptor 3 mutations and overexpression of ERB2, ERB3, and estrogen receptor. Then finally, these p53-like tumors, as I mentioned, are enriched with fibroblasts. They contain a SARC responsive signature that's been implicated in breast cancer bone metastasis and might be targetable. And finally, they might have features that suggest they're dependent on BCL2. And we have drugs that target BCL2 that are clinically available. So I'm going to skip the rest of these slides so that we have time for discussion. But to summarize what I've told you today, I think all solid tumors are being pulled apart in this manner and are being considered as multiple disease entities. And I think the striking biological similarities that we see between bladder and breast cancers really does mean that we can exploit the knowledge that's been gained in breast cancer research over the last couple of decades. They've been way ahead of us in funding and in research progress. We can just grab that and run with it as well. Really, essentially the same transcription factors that regulate basal breast cancer biology are regulating basal bladder cancer biology. So I think the future is actually really, really bright for bladder cancer research because of the genomics that's been performed. Because none of us would have expected that this would have been the case, but they looked a lot similar to other types of cancer. So with that, I'll be happy to stop and take any questions you have. Thanks.